Okay, welcome to Introduction to Bacteriology. I am Dr. Johnson. If you look at the first page of your handout, you'll see a summary of most of the items that you'll need to know for this lecture. Now, the modern study of bacteriology really begins with this man, Antony van Leeuwenhoek, who reported in 1683 that he had peered under his microscope and found some little animals. He called them animalcules, and his descriptions were quite amazing. For example, he said, "I then most always saw with great wonder that in the said matter there were many very little living animalcules, very prettily and moving. The biggest sort had a very strong and swift motion and shot through the water or spittle, like a pike does. The second sort oft times spun round like a top, and these were far more in number. From the mouth of an old man who'd never cleaned it, an unbelievably great company of living animalcules is swimming more nimbly than any I had ever seen up to this time. The biggest sort bent their body into curves and going forwards. Moreover, the other animalcules were in such enormous numbers that all the water seemed to be alive." Now, most of you know that the biological kingdoms can be broken down into prokaryotes and eukaryotes. The major difference is that prokaryotes have no nuclear membrane, nor do they have organelles. There is a nuclear region where you have the DNA, but there's no nuclear membrane as shown on the right. There's no Golgi, there's no rough endoplasmic reticulum, and there's no mitochondria. Now, you might be surprised that some bacteria do have electron transport chains. But where are these located if there's no mitochondria? They're located in the cytoplasmic membrane when present. Now, prokaryotes can also be divided into bacteria and archaebacteria. However, archaebacteria are not pathogens, so we'll not discuss them. So there's three major bacterial shapes. There's bacilli, which are rod-shaped, there's cocci, which are spherical, and there's spirilla, which are spiral. But there are many variations on the theme. For example, there's coccobacilli. There's thin, elongated, spindle-type rods, as shown here, typical of Fusobacterium. And there's different groupings, which are really caused by cell divisions in one or more planes. So there's diplococci, which are characteristic of Neisseria, Neisseria meningitidis, and Neisseria gonorrhea. There's chains, which are characteristic of streptococci. Streptococcus pyogenes, which causes strep throat, is an example. And then there's clusters. For example, the staphylococci, Staphylococcus aureus, is an example. Now, I'm listing the bacterial names just as a reference. You don't need to memorize them for this lecture. So bacteria have a cell envelope, and that envelope includes a capsule, if they do have a capsule, a cell wall, and a plasma membrane or cytoplasmic membrane. Now, some bacteria have two membranes. Now, way back in 1884, Hans Christian Graham noticed that most bacteria could be divided into two types, based, in fact, on whether they held onto a stain or lost a stain when they were treated with alcohol acetone. We now call these designations gram-positive and gram-negative in deference to him. Now, the gram stain will be important for your future because you will treat patients with particular antibiotics based on what the gram stain result is. And you'll be doing this in a laboratory. So first of all, you'll take a bolus of bacteria and you'll heat fix it on a slide. You'll then stain it with crystal violet. You'll wash that stain off, and then you'll add iodine, which forms a complex with crystal violet. You'll then wash the cells with alcohol acetone, and then counterstain them with saffron and red. Now, gram-negative cells will lose that first stain, and therefore, after adding saffron and red, they'll be counterstained red or pink. However, gram-positive cells will hold on to that original crystal violet and appear purple or violet in a gram stain. So what's going on here? Well, gram-positive cells have a very thick cell wall, whereas gram-negative cells have a very thin cell wall. Gram-negative cells also have an outer membrane. So what happens here is when the dye is taken in by both cells, gram-positive and gram-negative cells, and then fixed in this region here on the cytoplasmic membrane and also inside the cell wall, and alcohol acetone is added, the cell wall collapses inward because the alcohol acetone is a dehydrating agent. And that cell wall collapsing in gram-positive cells traps the dye in gram-positive cells, but the dye is lost in gram-negative cells because the cell wall is too thin to hold on to the dye. Not only that, there's an outer membrane that is dissolved by the alcohol acetone. So let's look at the cell wall. It's made of peptidoglycan. What is peptidoglycan? Well, you have glycan sheets made of N-acetylglucosamine and N-acetylmuramic acid. N-acetylglucosamine looks just like glucose, except there's an N-acetyl group on it. N-acetylmuramic acid looks just like N-acetylglucosamine, except there's a tetrapeptide hanging off one of the carbons. And that tetrapeptide can be cross-linked with a neighboring N-acetylmuramic acid tetrapeptide. So what causes this peptide link? There are enzymes called transpeptidases, and these enzymes are responsible for the cross-linking. And these transpeptidases actually bind penicillin and are inhibited, so they cannot cross-link in the presence of penicillin or other beta-lactam antibiotics. Because of that, these transpeptidases are called penicillin-binding proteins. Here's another cartoon of the peptidoglycan. Here's N-acetylmuramic acid forming a peptide cross-link with a neighboring N-acetylmuramic acid and N-acetylglucosamine. So here's a cartoon of a gram-positive cell wall that I drew. Notice there's multiple peptidoglycan sheets. Gram-positive cells can have up to 50 different sheets. But I also have breaks in these sheets. Why? Because as the cell divides and grows, it has to keep reforming this peptidoglycan layer. To do that, it has to use enzymes to break down these peptide cross-links, and then the transpeptidase goes ahead and remakes that. And the name of these enzymes that break it down are autolysins. So autolysins break it down, transpeptidases remake it. So what happens in the presence of penicillin? You add the penicillin and you cannot remake it. The autolysins start degrading the peptidoglycan and the peptide cross-links cannot remake it. And so the cell lyses, it explodes because there's a high osmolality inside the cell. Now gram-positive cell walls also have teichoic acids. These are absolutely unique throughout nature and are only found in gram-positive cell walls. These are polyribitol or polyglycerol phosphate polymers. Some of these actually contact the cytoplasmic membrane through a diacylglycerol attachment. And when they do, they're called lipoteichoic acid. Now these teichoic acids are negatively charged, so they can actually attach and bind to epithelial cells. You should note that when gram-positive cells lyse, they can release teichoic acids, and these teichoic acids are highly immunogenic. They can activate complement and also cytokines. So they can act like toxins, just as endotoxin in gram-negative cells does, but it tends not to be as toxic as endotoxin. Okay, here's another cartoon showing a better rendition of the peptidoglycan layers, more in 3 dimensions. So the individual N-acetylmuramic acids can connect to sheets above as well as within a sheet. So gram-negative cell envelopes are much more complicated because they have both an inner membrane, that's the cytoplasmic membrane, and also an outer membrane. The space in between the two membranes is called the periplasmic space. And the peptidoglycan can adhere to the outer membrane through these lipoproteins, as shown here. Now, the outer leaflet of the outer membrane has lipid A, which is part of a larger structure known as lipopolysaccharide. Now, about half of the number of the lipids in the outer leaflet are, in fact, lipid A. Now, lipopolysaccharides are highly negatively charged, and because of that, they attract a dense population of cations, such as magnesium and calcium. They're so highly dense that hydrophobic molecules that are grease-loving and should easily be able to get across the outer membrane cannot reach the outer membrane. Hydrophilic molecules can easily get past the cations, but they can't cross the outer membrane because they're not grease-loving. So how do gram-negative cells get nutrients? They get them through these porins, as shown here, here, and here. E. coli has about 400,000 porins for every cell. So small hydrophilic molecules can get across these pores by diffusion. But large hydrophilic molecules cannot. How large? Well, these porins are actually pretty small. They block anything larger than about 1,000 molecular weight for E. coli. But for some organisms, like Pseudomonas aeruginosa, they are very constricted. They're very small, and because of that, they have native antibiotic resistance. So many antibiotics are too large to get past the porins. The fact that gram-negative cells have both an outer membrane and also a very thin peptidoglycan layer means that they're not going to be susceptible necessarily to the same antibiotics that gram-positive cells will be susceptible to. So the lipopolysaccharide is very important because it is an endotoxin, and the toxic portion of the LPS is in fact lipid A, so that mice that are injected with just lipid A die from shock. Now you don't need to know the structure of the LPS, but you should know that there's three portions. There's the lipid A portion, which is the toxic portion. There's the core oligosaccharide. And then there's this O antigen. And these O antigens are highly variable, even within the same genus and species. For example, you may have heard of E. coli O157H7. The O157 is in fact the O antigen, specific for that toxic strain. So what happens with the endotoxin is that lipid A binds toll-like receptor 4, which activates cytokines and platelet activating factor, causing activation of complement and coagulation, leading to endothelial damage. So symptoms include fever, which we can treat, hypotension, which we can treat, but intravascular coagulation prevents blood flow, and this leads to organ failure. And just 1 microgram per kilogram can induce shock. And here are a couple of examples of disseminated intravascular coagulation from Neisseria meningitidis. On the right is an 8-month-old Indonesian child, and you can see the dead tissue on his legs and feet. The only way to treat this is to debride, that is to remove all the dead tissue. Fortunately, this child did not have to have his legs amputated, and he survived. Unlike this woman on the left, who had to have her fingers amputated due to disseminated intravascular coagulation. All right, let's switch gears to cover a couple of microbes that don't gram stain very well. There's Mycobacterium tuberculosis, the cause of TB, and Mycobacterium leprae, the cause of leprosy. These organisms have a very thick, waxy cell envelope, as shown here, and this is due to mycolic acid, which is 60 to 90 carbons in length. So you can imagine how incredibly hydrophobic this would be. So this actually inhibits dyes from the gram stain from crossing it. In fact, when we do stain it, we use an acid-fast stain. And to get the dye in, we have to use tricks. We have to heat it up or add quite a bit of phenol to get that dye in. Once you wash out the dye, however, that dye stays in the cell no matter what. It holds on fast to that dye even in the presence of 3% hydrochloric acid. And that's why we call it an acid-fast stain. Now, in contrast to the mycobacteria, the mycoplasma have no cell wall. Don't confuse mycoplasma with mycobacterium. So mycoplasma have no peptidoglycan. Their membranes are stabilized by sterol. Just like humans. Humans have membranes that are stabilized by cholesterol. Mycoplasma can have cholesterol in the membrane, but it has other sterols as well. And even though it has sterol to stabilize the membrane, they're very osmotically sensitive. But one of the main things you're going to need to know when you're on the wards is that mycoplasma do not care about beta-lactam antibiotics. They resist them. Why? Because beta-lactam antibiotics like penicillin inhibit peptidoglycan synthesis. Mycoplasma has no peptidoglycan, so it could care less whether you inhibit peptidoglycan synthesis. An example of mycoplasma is Mycoplasma pneumoniae, which causes walking pneumonia. So bacterial motility is a virulence factor. Motility is driven by rotary motors, and these rotary motors are driven by ion motive force, either proton motive force or sodium motive force. So, for example, protons that have been kicked out by the ETS come back in and they cause lateral torque like a windmill on this motor. And so just like air going through a windmill, the motor spins. So here's 4 different E. coli strains swimming in real time through liquid. I took this video myself and noticed that these organisms swim at about 30 microns per second. Well, what about spirochetes like Borrelia burgdorferi? They have a motor on each end of the spirochete, and the flagella is in between the inner and outer membrane. The outer membrane has been removed so you can see it. So it's an endoflagellum. And these organisms can move through very viscous environments. For example, here's a real-time video on the upper right here of Borrelia burgdorferi, which causes Lyme disease, moving in real time through mouse skin. It doesn't seem to care whether it's highly viscous or not. Now, this film has not been published yet. I got it courtesy of Mark Wooten, who's a colleague of mine from University of Toledo. Now on to fimbriae. These are highly sticky projections. They bind surfaces, and once they attach to the surface, they are relentless. They do not let go. They set up shop. The cell starts dividing, becomes 2, 4, 8, 16, and so on, and causes an infection. So a urethral or bladder infection by E. coli absolutely depends on the presence of fimbriae. Same thing with Neisseria gonorrhea. Its fimbriae stick tenaciously to the urethra, and you cannot get rid of this no matter how hard you urinate. Now, you may have heard the term pili or pilus. These are the same things as fimbriae. Pili were discovered in the US, fimbriae were discovered in the UK and reported, and they turn out to be the same thing. So you'll hear these words used interchangeably. However, typically the sex pilus is specific for conjugation in bacteria. So the sex pilus is a fertility pilus that's involved in mating between bacterial cells. So here's the fertility pilus here. We used to think that the DNA was transferred from one organism to another through this. But we now know that this pilus is really thrown out like a fishing rod, and it attaches to a surface or another organism, and then the bacterium that has the pilus reels the other bacterium in for conjugation, which occurs when they have contact. Now, as I alluded to, the pili can actually be used for locomotion. So if you look in the upper video on the right, you can see pili retracting. And this occurs by depolymerizing subunits into the cell. Now that video is sped up, as is the next one on the lower left. You can see motility on the one on the lower left. So the fishing line's thrown out, it attaches to a surface, and then the bacterium pulls itself forward. Now, capsules are a very important virulence factors because they protect the bacterium from the immune system. And with Streptococcus pneumoniae, for example, the virulence increases 1,000-fold if it's encapsulated. If you look on the left panel, you'll see 2 mice. The upper mouse and the lower mouse were injected with exactly the same amount of Streptococcus pneumoniae, but the upper one was injected by a non-encapsulated one. The lower one had the capsule. You can look under the microscope and you can use stain to see the capsule. So the capsule itself isn't stained, but for example, India ink can perfuse through that capsule and stain the organism, as shown here. This is Streptococcus pneumoniae, which forms lancet-shaped diplococci, as shown here and here. So what's going on here? Well, normal phagocytosis by a macrophage can be seen here. In the lower center, you see a macrophage, and it senses all the bacteria. And notice this is sped up, but it circumscribes all these bacteria, and it starts digesting them. And then it'll move on to another set of bacteria. And this is what you want. And this is what you'll get from unencapsulated organisms. So, for example, in the upper right panel, here's E. coli without a capsule, and it's helpless against a phagocyte. Now, if we look at the lower left panel, Streptococcus pneumoniae, it's just pushed around by the macrophage. It's poorly opsonized. It's difficult to opsonize capsulated organisms because they are usually polysaccharide, and polysaccharides are poorly immunogenic. So here's an example of a thick capsule, as shown here, but actually not all capsules are alike. Some organisms have a very thin, loose network of fibers instead of the tight capsule. And this network is called a slime layer, and it's very sticky. It can actually be used in attachment, which occurs with Streptococcus mutans, the cause of dental caries. Although Streptococcus mutans also has fimbriae, and fimbriae are responsible for initially setting up the attachment. If you grow organisms that have a thick capsule on a petri dish, you can usually tell that they have a capsule because they have a glossy appearance, as shown here, here, and here, particularly for Klebsiella pneumoniae. Some of you are going to have Streptococcus pneumoniae as your unknown, and I'll come around in the lab and I'll look at your plate and I'll say, wow, look at this. Does that look a little shiny? What does that suggest to you? And you'll scratch your head and wonder why I'm asking. Well, it means that you have a capsule. So organisms that produce exopolysaccharides tend to form biofilms pretty fast. So biofilms are caused by slimy bacterial exopolysaccharides, plus other host factors like proteins and DNA. So here's an example of a biofilm forming just hours after debriding a tooth. So it grows in this fissure, and this biofilm can help protect the organism against host factors such as the immune system and also antibiotics. And biofilms are the bane of infectious diseases. So they form everywhere. Here's a heart valve, catheters, urinary catheters, vascular catheters, voice prostheses, implants. Here's a 3-day-old sock. Well, our body does have some antibacterial agents at the forefront of the fight. For example, both tears and saliva have lysozyme. So lysozyme is antibacterial. It digests the cell wall. What does it digest? It actually cleaves the sugar bond. It cleaves between N-acetylglucosamine and N-acetylmuramic acid. This differentiates it from the beta-lactam antibiotics, such as penicillin, which don't cleave anything. They just bind the transpeptidase and prevent cross-linking between N-acetylmuramic acids. And remember, it's autolysins, not penicillin, that actually breaks this bond while the cell is growing. So if you add penicillin to actively growing cells, this is what transpires. The cell lyses because of the high osmotic pressure inside the cell. So to summarize, penicillin blocks peptide cross-linking by binding the transpeptidase. It doesn't cleave anything. Lysozyme cleaves the sugar bond between N-acetylglucosamine and N-acetylmuramic acid. Lastly, there's endospores. Endospores are very hardy forms of bacteria that are difficult to kill. They're found in both Bacillus and Clostridium genera. So these organisms form endospores in response to unfavorable growth conditions, typically starvation. And once they form spores, they can live in nature for decades, if not hundreds of years. You can even throw them in boiling water, and you will hear them sing, "Boiling water never bothered me anyway." So they're very hardy. So why are they so hardy? Because they form this very thick calcium dipicolinate shell. Now on to a little bacterial physiology. So bacteria have different degrees of fondness for oxygen, and that actually determines where you'll find these organisms on your person. So organisms that are found in the gastrointestinal tract tend to be different from those on the surface of your skin. First of all, there's obligate aerobes. So like humans, they have an electron transport system that reduces oxygen to generate ATP. If you were to throw these organisms in a test tube with nutrient broth, they'd appear like panel A. They'd be growing near the surface of the nutrient broth. Then there's facultative anaerobes. They can generate ATP with or without oxygen, but they still prefer oxygen. So if you put them in nutrient broth, they would form a growth distribution as in panel C, as shown here. Most of the organisms would be near the surface. Then there are microaerophiles, which require oxygen, but they can't handle much of it. They require a low amount of oxygen. And if you were to put them in a tube of nutrient broth, they would form near the meniscus, but not up to the meniscus, as shown here in panel E. Then there are anaerobes. There's obligate anaerobes and aerotolerant anaerobes. Obligate anaerobes can't use or tolerate oxygen at all. They can't tolerate reactive oxygen species such as hydrogen peroxide or superoxide. And this is produced in metabolism with oxygen. So they like to ferment, and during that process they tend to form stinky, volatile sulfide products. And so you'll find these in your colon and also in your mouth and crevices when you wake up in the morning as you sleep throughout the night. Then there are aerotolerant organisms. They can tolerate oxygen but can't use oxygen. These organisms tend to have a pretty good supply of catalase or peroxidase to handle the hydrogen peroxide and superoxide dismutase to handle the superoxide. So now on to methods of energy generation. As I mentioned earlier, the cytoplasmic membrane of many bacteria have an electron transport system. And just like the electron transport system in humans, many bacteria can take NADH and reduce oxygen to water to generate ATP. So we start out with NADH, which has a very low redox potential of a minus 320 millivolts, And oxygen, which has a redox potential of 820 millivolts, so it is the Arnold Schwarzenegger of electron acceptors, and it pulls these electrons through the electron transport system at the same time pumping out protons. And the protons come in through an ATPase. The ADP condenses with phosphate to form ATP. So that's fine and good, but many bacteria actually have other electron acceptors they can use other than oxygen. So instead of using aerobic respiration, they use anaerobic respiration. And I know it sounds like an oxymoron. How can we have anaerobic respiration? Well, respiration is simply using the ETS to generate energy. Aerobic means using oxygen, so this is anaerobic respiration. And some of these alternative electron acceptors include nitrate, fumarate, even iron. None of them have a redox potential as high as oxygen, so they can't generate as many ATPs per electron going through the electron transport system. Notably, pathogens like E. coli, members of the Enterobacteriaceae, all can reduce nitrate to nitrite. And because members of this group are known for urinary tract infections, particularly E. coli, so if you go to your corner pharmacy, you can get a dipstick that actually has a test for nitrate on it. So you take the dipstick and you put that in a stream of your urine, and if it turns a particular color, it means that nitrite has been converted to nitrate, and you likely have an infection with a member of the Enterobacteriaceae, particularly E. coli, in your urinary tract. Lastly, fermentation. This is kind of a modification of anaerobic glycolysis, so we actually can ferment. During a sprint, when oxygen can't get to the scene fast enough, we convert glucose to pyruvate, and then to reoxidize the NADH, during the substrate level phosphorylation cascade, we convert pyruvate to lactic acid, and that reoxidizes NADH, so you can get more NAD for the pathway. So many bacteria ferment exclusively, and they're not wedded to glucose going just to lactic acid. There's all kinds of other products that can be produced, such as butyric acid, propionic acid, butanol, ethanol, and the like. Now, some bacteria can't generate enough ATP to survive by themselves. They become obligate intracellular parasites. Now, you don't need to remember the names of these right now, but you'll be learning about them throughout the year. But these include Chlamydia trachomatis, which causes trachoma and blindness, and also sexually transmitted infections. Rickettsia rickettsii causes Rocky Mountain spotted fever. Coxiella burnetii causes Q fever, and Ehrlichia causes ehrlichiosis. The important point here is the diagnosis can be difficult. Why? Because when you take a specimen of the discharge from humans, you may not see any microbes at all because they'll be intracellular. So you actually have to scrape off cells that might have the microbes inside them. So a very important element for bacteria is iron. And free iron can be very difficult for a pathogen to find in the human body because it's tied up with transferrin. But bacteria have their own type of transferrin. They're called siderophores, and they bind tenaciously to iron. They form a complex that is actively transported into the cell. So because iron is such a difficult commodity for many pathogens, if you take iron pills or have an iron stores disease, it can increase the pathogenicity of the pathogen. As I mentioned before, reactive oxygen species are toxic to cells. They're produced in the metabolism of oxygen, and these include superoxide anion, hydrogen peroxide, and also hydroxyl radical. And so bacteria living in an oxygen environment produce superoxide dismutase to handle the superoxide anion, and catalase or peroxidase to protect them from hydrogen peroxide. Well, you're going to learn how to do a catalase test in the lab. And this catalase test discriminates staphylococci from streptococci. These are major human pathogens. So the staphylococci are catalase positive. Catalase converts hydrogen peroxide to water and oxygen. So if you take a bolus of cells from a petri dish and you put them in hydrogen peroxide as shown here, you'll see bubbles of oxygen. Now this happens to be Staphylococcus, which has a lot of catalase. There are some catalase positive organisms, for example E. coli, where you won't see nearly this many bubbles. Now, an easy way to remember that Staphylococcus has catalase is you can see a CAT in the name, cat, for catalase. Streptococci does not. There's no A, so you don't get cat out of it. Another important test that you'll be doing in the lab is the oxidase test, and this tests for cytochrome C oxidase in the electron transport chain. In this test, cytochrome C oxidase rips off electrons from a colorless dye known as TMPD. But the oxidized form of this dye turns blue. What you'll do is remove cells from a plate and scratch it onto this grid here and wait 20 seconds. If it turns blue within 20 seconds, it means it's oxidase positive. Now, this is a very quick way to differentiate certain microbes like E. coli, which is oxidase negative, from Pseudomonas aeruginosa, which is oxidase positive. And notably, these two look very similar under the microscope. They're both gram-negative bacilli. Now a few words about the exponential growth of bacteria. So bacteria grow by binary division. So one cell becomes 2, 4, 8, 16, 32, 64, 128, 256, 512, 1024 in just 10 generations. So if you count it on your fingers, you know that that was just 10 generations. So it goes from one cell to roughly 3 orders of magnitude higher in 10 generations. That means that in 10 generations, one cell becomes 1,000, 20 generations 1,000,000, 30 generations 1,000,000,000, 40 generations 1,000,000,000,000, and so on. Well, E. coli can double in 20 minutes. That means it can go from one cell to 1,000 cells in just 3 1/3 hours. And if growth continued, E. coli would reach the mass of the Earth in just 45 hours. Of course, it can't grow to that mass because it would run out of nutrients. An important point here is that you can plot this exponential growth to determine the doubling time. If you use a linear scale, you're not going to be able to do this because it's going to rise rapidly in an exponential format. But if you use a semi-log plot, which is logarithmic on one scale, in this case the y-axis, and a linear plot on the x-axis, you will get a straight line. So imagine starting with just 10 cells. 10 goes to 20 in one generation. 20 goes to 40 in another generation. 40 goes to 80 in the third generation. 80 goes to 160 in the fourth generation, and so on. And so all you need to do to determine the doubling time of a bacterium is to dilute some bacteria out into nutrient broth and then start growing it. You take a sample out, put it in a spectrophotometer every once in a while, and then you look to see how dense the population is. And then you plot that, and you look, for example, as to where the line crosses 20, 40, 80, and so on, and you can determine the doubling time. Of course, it's not going to start out in an exponential phase. There's a lag phase. Why? Let's say you take a throat specimen from someone and put it in nutrient broth. Well, that's going to be in a different environment, so the bacterium has to figure out what transporters to make, what enzymes to make, and all this takes some time as it starts inducing and repressing certain genes. Eventually, it starts growing faster and faster, and it reaches the exponential phase as shown here. And then, of course, it runs out of nutrients, so it reaches a stationary phase where it relies on intracellular stores. Eventually, it will exhaust those intracellular stores. Autolysins degrade the cell wall, the cells lyse, and you have a death phase where you have an exponential decline in bacteria. Let's finish with a few words about the human microbiota. Microbiota includes all microbes on or within humans. We used to call this the human flora. In contrast to the microbiota, the microbiome includes all genes within those microbes in the microbiota. So humans have between 2 and 6 pounds of microbes living on and within their body. And these have specific roles that help us. So for example, normal colonic bacteria produce vitamins. They aid in digestion. Negatively, they do produce flatus. But on the positive side, they elbow pathogens out of the way, and they help develop our immune system. Now, over recent years, we've learned that disruption of the colonic bacteria, for example with antibiotics, can actually allow pathogens to grow. And one of these pathogens is Clostridioides difficile, which can be found in many people's colons in the form of spores. And the normal microbiota prevents those spores from germinating. However, if a lot of this microbiota is killed off, for example with antibiotics, these spores can germinate, causing ulcerative colitis. And these infections are very difficult to get rid of. Under extreme cases, we found that fecal transplants can actually help. Other recent studies have shown that an imbalance in the microbiota in the gut can lead to irritable bowel disease, cause allergies, diabetes, and even CNS changes like depression, anxiety, and obsessive-compulsive disorders. And this will be a fertile area of research for years to come. And with that ends the Introduction to Bacteriology lecture.